All right, we are back live and we are ready for the next speaker, Arezoo Adrekani, who from Purdue University, and she's a recipient of a 2020 Young Investigator Medal. And the title of our talk is Transport of Particles, Drops, and Organisms in Density Stratified Fluids. Right, so the floor is yours.
Thank you very much, Stefano, for the introduction. And it's a great honor to receive SES Young Investigator Medal. And I would like to thank the organizers as well as the award committee. I would like to also thank my nominators and mentors who've had great influence on me over the past several years, as well as great PhD students I've worked with, which had led to these accomplishments. The reason we work on motion of particles, drops, or organisms in density stratified fluid is that it has been shown that intense biological activity and accumulation of particles and organisms are associated with pycnoclines. These pycnoclines are regions where we have sharp change in density, either because of variation in temperature or salinity. For example, these measurements in Pacific Ocean shows sharp change in density, and exactly where we have this change in density, we see sharp increase in concentration and mean volume of marine snow particles. These particles are made of organic matter that are basically made of dead parts of organisms attached together by their mucus, and as they settle down to the deep ocean, they affect carbon cycle. There are also large-scale environmental processes that are correlated with seasonal stratification. For example, phytoplankton bloom, these greenish color here, their formation is correlated with seasonal stratification, and they consume CO2 and they die, they sink to the bottom of the ocean. So it's a natural way of carbon sequestration. It's referred to as natural biological pump. In fact, this phytoplankton produce half of the oxygen on Earth's atmosphere, more than terrestrial plants. But they can be toxic, and when they're toxic, like blue-green algal bloom, they can disrupt the water supply system. They can lead to livestock perishing, and Australia holds a record information of these. In 1991, thousands of kilometers of Barun and Darwin River were affected. But also, you may remember in 2014, Lake Erie had one of these red tide or algal bloom, and the water supply system was disrupted for Toledo and some of the other cities. Not only at the scale of these larger scale environmental processes, the stratification matters, but also at the scale of individual organisms. Some species of krill cannot swim through thermocline, or some species of dinoflagellates cannot swim through holocline, which is when we have a stratification because of change in salinity. As a hydrodynamicist, what is interesting to me is that how much fluid dynamics matter versus how much biology contributes. When I started this field a few years ago, the first question I was getting from other researchers was, these particles and organisms are too small to be affected by stratification. Because there is a length scale in the problem which is related to change in density over the density gradient, and that's on the order of meter. But what I've shown is that there is another length scale which is related to viscosity, diffusivity, strength of stratification, and gravitational acceleration. And for example, for measurements I showed earlier for Pacific Ocean, that can be as small as few hundred micrometer or millimeter. So even though small bacteria or smaller plankton are not affected, but larger plankton, zooplanktons, marine snow particles are affected by density gradient. So the simplest problem we can start with is the solution of a point for singularity in a stratified fluid. The solution for a point for singularity in a homogeneous fluid was known for centuries, and people have used it. And because the governing equations are linear in a Stokes regime, we can actually use the superposition to look at more complicated problem. So what I started looking at is, what if we have this point for singularity in a linear background density? Can we look at the solution? And we can linearize the equation by neglecting inertial effect and convection effects. And basically, we have the momentum equation, we have the continuity equation, and we have a convection diffusion equation for temperature. And temperature and density are linearly related to each other. Now, if we linearize the equation around the basis state corresponding to linear density stratification and the hydrostatic pressure associated with that, we end up with three linearized equations for pressure perturbation, velocity, and density perturbation, which if you go to the Fourier space, you can algebraically solve for them. So the only remaining step is then to semi-analytically or numerically calculate the Fourier inverse and get the solutions in the physical space. So let's see how the solution look like. The far field solution of a settling particle is represented by a point for singularity. And the solution, the flow field is downward everywhere in a homogeneous fluid, but in a stratified fluid, we see formation of these toroidal eddies, which are present because the stratification suppresses vertical motion of the fluid. Now, if you look at the point for singularity between two walls, which also suppresses vertical motion of the fluid near the wall, it's qualitatively similar, you see these toroidal eddies. If you're interested in solution of a microorganism, then that can be modeled as a force dipole, because there's a thrust generated by the flagella, and there's a drag force acting in cell body. And this force dipole in a homogeneous fluid generates a radial flow, which decays as 1 over r squared. But in a stratified fluid, we see formation of these toroidal eddies. But how does the stratification can affect life of these organisms? If you look at the velocity disturbances generated in stratified fluid normalized by the one in homogeneous fluid, we see velocity decays much more rapidly and is suppressed. That can affect life of these organisms in different ways. One way is that many of these organisms, like copepod, detect their predator or prey using mechanosensing. So they have these antennae, and there are these little hairs on those antennae which detect flow disturbances. So you can quantify that now that we have this semi-analytical solution by calculating the detection volume inside which flow disturbances exceed the threshold. And if you do that, you see that the detection volume scales with the size of the organism over this length scale of a stratification to the power minus third. You can also represent it in terms of Rayleigh number. So if you do now calculation for 600 micrometer prey, then there's a 50% reduction in detection volume. So mechanosensing is suppressed. So perhaps these stratified regions act as niches where these organisms can hide. But energetically, it's also more expensive to swim across these pycnoclines, because they not only need to overcome viscous dissipation, but also they need to mix fluid around them. And that extra energy they need to spend also scale with Rayleigh number to the power 3/4. There are older experiments that support the fact that density gradient matters and not the biology, in the sense that they did experiments for different zooplankton and mollusk and other organisms, and they're homogeneously distributed in a homogeneous fluid. Each little pixel corresponds to an organism. But then they added fresh water on top of salty water, which is basically the organisms are swimming, and they swim primarily near the interface. To test whether density gradient matters or they like salt gradient, they added more sugar to the top, so density is uniform, but you still have the sharp gradient in salt as well as in sugar, and they went back to the homogeneous distribution. Our own experiments with Sigma akoshibo, which is the algae cell leading to red tide, shows the same thing. So if you have a salty water in the bottom and you add fresh water at the top at time 0, in about 10 minutes, you see them accumulating near the interface. So there are also other effects that are happening because of the climate change itself. The climate change, the effects of global warming and ocean acidification, you have heard a lot probably. But the one that you may have not heard as much is ocean deoxygenation. And there are some regions in the ocean that have lost 40% of their oxygen. The volume of water containing zero oxygen has more than quadrupled. And the reason for this are threefold. One is solubility changes. So water gets warmer and it can dissolve less oxygen. But another one is related to stratification, in fact, because the climate change has led to more, has enhanced the stratification in oceans and seas. And that changes the dynamics of these organisms, as well as the oxygen distribution, in the sense that the top 200 to 1,000 meters of water is well mixed because of wind and tide and other effects, and that is rich in oxygen as well as in nutrient. But the bottom layer is has much less oxygen and less nutrient, and the stratification has enhanced over the years, and that is reducing the mixing to the deep ocean. Another effect is due to change in metabolic rate by these organisms, which just 2-3% degree warming can increase their metabolic rate by 30 to 50%, and consequently leading to larger scale ocean hypoxia. So the solution I talked about assumes we have zero Reynolds number, or we can neglect inertia, and we can neglect convection over diffusion. But if you are interested in larger inertial regime for organisms or particles, we need to look at it numerically. If we look at the single particle settling, the flow field behind the particle is downward in homogeneous fluid, but in a stratified fluid, there will be a jet of light fluid that is upward. And if you look at the settling velocity of the particle in a homogeneous fluid, it reaches a terminal velocity, but in a stratified fluid, it reduces significantly, and it can even oscillate. The particle itself could oscillate, and the oscillation itself is correspond to what is called Brun-Weisel frequency, which is the natural frequency of the oscillation of a fluid particle displaced from its neutrally buoyant level. For this larger inertial regime, fluid number is a relevant parameter to look at, which is the ratio of inertial force to buoyancy force. So if you look at the particle in a homogeneous fluid, it has this formation of vorticities around it. But in a stratified fluid, we see formation of these secondary vortices and tertiary vortices and so on and so forth. And the reason is that if you look at the vorticity equation, there is this baroclinic term that is now non-zero in a stratified fluid. Basically, behind the particle, you have gradient in density that is horizontal and gradient in hydrostatic pressure or is vertical, and the cross product leads to this formation of secondary vortices and slows down the motion of the particle. Looking at the velocity contours, we can also distinguish the Lie waves that forms behind the particles, and it slows down the particles further. In many of the examples I talked about, we don't have an isolated particles, the particles are interacting. So in a dilute regime, perhaps pure particle interaction is the most dominant. So if you have two particles interacting in a homogeneous fluid, they go under what is called drafting, kissing, and tumbling, because the top particle moves faster, and then at some point it collides with the bottom particle and then tumbles and separate. So if you look at velocity as a function of time, you do see this jump in velocity when they collide, and finally they separate. As you increase the stratification, then not only the settling velocity of these particles are slowed down, but also their separation distance is less, and they tumble far less. And in fact, there's a critical fluid number below which, or there's a stratification beyond which, you do not see any tumbling. So rather than seeing drafting, kissing, and tumbling, you would see what I call drafting, kissing, and separation. The particles do not tumble, they collide and separate and eventually move together if the stratification is strong enough. For particles side by side, the vorticity of one particle is blocked by the presence of the other particle, so they are separated from each other. But in a stratified fluid, they move together. So initially they separate because of larger inertia, but when they slow down, they move together and aggregate. What about non-spherical particles? Many of the particles in oceans or lakes are not spherical. In a Stokes flow, we know that the angle is indeterminate, meaning that particle keeps whatever initial orientation it has. In inertial regime, it would reorient so it settles on its broadside. What about in a stratified fluid? Is it different in low or intermediate Reynolds number? And we did both experiments as well as computations for that. For experiment, we built a linear stratified fluid with constant n, and using a double bucket system. And we observed three phases. So initially, the particle is settling on its broadside. Then later on, we have change we have an instability. The particle settles on H-wise, and when it gets to neutrally buoyant level, it will reorient and settle, and it becomes broadside again. This video, I believe, doesn't run, but basically we see exactly the same thing in numerical simulation. Here we are showing vorticity and here velocity field, where we do see these internal Lie waves forming. And also we do see initially the particle keeps the broadside and settling, and eventually settle on its H-wise direction. If you look at the initial stage of the settling, which is a quasi-steady behavior, the reason we have this enhanced drag is the particle will trap a region of light fluid around it from upper layer, and that buoyancy force acting on it leads to enhanced drag. So that would depend on the thickness of this shell of light fluid, which scales with the length scale over which viscosity diffuses over the time scale of a stratification, as well as this distance that the light fluid has trapped, which itself is dependent on fluid number. So if we look at this enhanced drag in a stratified fluid normalized by the one in homogeneous fluid, it depends on the buoyancy force acting on shell of light fluid that is normalized by the drag acting on the particle. And because of what I just explained, we would be able to find actually that relationship. And what we found is that it very well described the experimental and computational data we have for the drag as we predicted with this analysis. The next phase is the instability of broadside unsettling. And the reason is that we get this jet of light fluid behind the particle, which the point at which this jet is separated from the particle tilts. It's not center of the disk anymore, and that buoyancy-induced torque leads to change in orientation of the particle. And the Archimedes number, which is defined as the basically dimensionless parameter that is defined as explained here, would depend on the stratification length scale that I talked about earlier. So this is stratification length scale, which is basically where buoyancy effect and viscous effect balance each other, play an important role in describing the dynamics of the particles as well as the organisms. If you have swarm of these particles or drops, these are now drops that are lighter than fluid around on them. So this, not only the rising motion is slowed down, but also they are much less dispersed compared to homogeneous fluid. The slowed down motion can be quantified by looking at the Reynolds number, and this stratification reduces the rising Reynolds number for single drop as well as suspension of drops, but is much more effective in reducing the rising motion for suspension. And the velocity fluctuations are also suppressed because the stratification suppresses the vertical motion of the fluid. The clustering can be quantified by pair correlation function by finding the probability of finding a droplet at angle theta and distance r from another particle. And if we plot this pair correlation function as a function of radius, we see a peak in homogeneous fluid. So still, there is already some clustering, but the peak is much larger when we have a stratification. And there's a peak at 90 degrees, but because there is a horizontal clustering in homogeneous fluid as well, but the peak gets larger as we increase the stratification. When we think about motion of particles, another effect to consider is the mixing they generate. So if you look at settling of a sphere, or a single sphere or multiple spheres, we do see these oscillations behind because of and the frequency depends on Brunt-Väisälä frequency. And this mixing can be quantified using drift because these particles trap these light fluid around them and they settle down. But when we look at cylinders, another mechanism come into play, and this mixing is enhanced, which I'm not going through the detail here, but what we see is the vortex shedding is very strong, and that itself leads to additional mixing for cylinders. Next, I'm going to switch gears a little bit. Still, I'm talking about a stratification, but now we have sharp stratification rather than linear stratification. So we have interface, either liquid-liquid or gas-liquid interface. And we want to know what happens to the dynamics of microbes or microorganisms near these interfaces. The motivation for this comes from the Deepwater Horizon oil spill, where millions of barrels of oil was released into the Gulf of Mexico. And microbes were the first responders. So if they were not present, the catastrophe would have been even worse. So microbes decompose much of the soluble as well as insoluble components of the oil. So the question we want to look at is that how hydrodynamics play a role in interaction of these microbes and oil droplets. There are already some experiment in the literature which shows nanorobots near polystyrene particles can get trapped. But can we see the same thing for bacteria near oil droplets? And if we do, how do surfactants affect it? The reason we're interested in looking at the effect of surfactants is that after the Deepwater oil spill, 7 million liters of dispersants or surfactants were released into the Gulf of Mexico with the hope of increasing that bioremediation. But what are the effects that these surfactants can really have? So we modeled this using the forced dipole, which I talked about earlier, to look at the bacteria, and we put it in near a surfactant-laden droplet, and we assume inertia is negligible. Forced dipole is a good approximation for the flow field. This is experimentally measured flow field around E. coli, and this is a numerical. This is a theoretical solution for a forced dipole. They are close to each other, and now what we can do is first find the solution to point for singularity near a surfactant-laden droplet, and then from that, then we can get higher order singularities like forced dipole and other singularities. And that leads to hydrodynamic-induced velocity we get and angular velocity we get, which governs the motion or trajectory of the microbes. So there is this hydrodynamic attraction to the droplet, which we can calculate using Faxin law. Knowing the image singularity, we need to put inside the droplet to satisfy the boundary condition on the droplet. And there's also swimming velocity for these bacteria. There are 4 dimensionless parameters to describe their motion. One is dipole strength, alpha. The other one is the radius of the droplet to radius of the microbe. The third one is the viscosity ratio of the droplet surrounding. And last one is the surface viscosity to exterior viscosity. And with that, we can find the deterministic motion of bacteria. So let's say first we look at single bacteria near a droplet. We see it, if the droplet is small, the bacteria get dispersed, but if it's large, it gets trapped around the droplet. And this is all hydrodynamic trapping. We could look at this trapping radius for different scenarios. It decays as 1 over alpha square, which is the dipole strength, though hydrodynamic matters a lot, but also viscosity ratio is important. So for rigid sphere, for example, trapping is more, hydrodynamic attraction is more, so trapping radius gets less. If you add a surfactant to it, we see a non-intuitive effect in the sense that surfactant leads to even more attraction near droplets compared to rigid particle, which is hard to explain. So in order to understand that, we did some experiment to see if the same thing happens, and I'll show that in a second. But we can also add in addition to deterministic motion, we can look at the stochastic motion of these bacteria, including run and tumble, as well as the diffusive motion. And we see if we have suspension of these bacteria, near clean drop, some of them get trapped, some get scattered. But when we add surfactant, we have much more trapping compared to clean droplets. So in the experiments, rather than looking at bacteria motion near oil droplet, we did flat interface. So we had a glass slide on the bottom. We had either liquid or gas-liquid interface, and we looked at the culture of bacteria like Pseudomonas butida as well as E. coli in the middle, and looked at their distribution. So we basically looked at the Z-scan used the Z-scan of the microscope and counted the number of bacteria at each plane to see their distribution. We see this peak near the surface, which comes also from hydrodynamics. That's due to image singularities that you need to use to satisfy the no-slip boundary condition on the surface of the solid wall. But near a CO2 liquid interface also, we see an increase in concentration, and it's even larger than solid surface. And that's in agreement with the theory I showed earlier when surfactants are present. The reason we use CO2 rather than air is because some bacteria have oxytaxis, they like air, so we wanted to separate the two effects, so we use CO2 here. And we use that theory to build a Brownian dynamic code where we include this run and tumble for bacteria, we include the all their hydrodynamic interaction, and then the results from this calculation fits very well with experiments we get from different liquids like dodecaine, mineral oil, soybean, and all the other liquids. We can also capture this circular motion near interface as well as the wall, and this is the PDF of that radius of curvature of the circular motion we get near bacteria, near the surface. What I talked about here was looking at hydrodynamic interaction, but hydrodynamic interaction is not the main mechanism that bacteria come to the oil. The main mechanism is due to chemotaxis, is an active mechanism where bacteria sense and direct chemical gradients. For example, for E. coli, the way it works is that it has multiple flagella. They are connected through motors to the cell body, and they form this flagella bundle. And once in a while, one of these motors rotates the other direction. They unbundle, it reorients into a new direction. And by changing this runtime and tumble time, they can go from region that they have less food to the region they have more food. And chemotaxis is basically what brings bacteria near the oil droplet, and then hydrodynamics needs to kick in to get it trapped near the oil droplet. So the stronger chemotaxis, more accumulation near the oil droplet, and weaker chemotaxis, we have more accumulation near the drop. But what is interesting is that in all this dynamics, hydrodynamics, which is described here with dipole strength, is very important, and we get significantly larger trapping for cases with larger hydrodynamics effect and larger dipole strength. So what is shown here is the time these bacteria remain trapped compared to the simulation time. So I'm going to summarize the talk. So in the first part, I talked about how mechanosensing is suppressed in the stratification, how motility and settling dynamics is reduced, and how attraction between particles is enhanced, and mixing can be caused by drift and vortex shedding. And in the second part, I would like to end up with a limerick I wrote for it, which is thanks to hydrodynamic interaction, surfactant help increase bacteria fraction on consuming oil droplets, eating them like chocolates as it's combined with chemotaxis reaction. And with that, I'm going to end, and please let me know if you have any questions.
Thank you, Arezoo, for the nice presentation. And congratulations again for your Young Investigator Medal.
Thank you.
Let's see if there are any questions by especially the fluid mechanics people attending. Update the screen. Yes, there's a question by Suni Jung. Says, nice talk. When a bubble or drop rises up in a stratified fluid, does the bubble deformation different from the one in homogeneous fluids? If yes, then how are they different?
Thank you, Sunny, for the question. Yes, it is different. So what we see is that for the droplet in homogeneous fluid, it's deformed much more, and that the main reason is that the droplets are slowed down in a stratified fluid. So the inertial effect is less, and their deformation is also reduced because of that.
All right, that was good. Let's see if there's anything else. I have to keep refreshing the page. Let's see. Well, I do not see any other questions at the moment, so I speak slowly in case people change their mind. Otherwise, I think we can thank Arezoo one more time, and again, congratulate you, and move on to the next and last speaker. Thank you.
Okay. Thank you very much.